So my perspective, I'm a chemist. I'm organic chemist, peptide chemist by training. I did all my degree in the Hebrew University in Jerusalem. I combined chemistry, biochemistry, microbiology along my academic journey. And in these days, we established a consortium, a huge project with Singapore. I will share also my colleagues from Singapore. And the idea of the project is to design bioengineering tools for next generation cellular agriculture, not particularly just on cultured meat, but on overall the idea of the cellular agriculture. So as we all will know and hear in this talk or working on this field, we have the main bioreactors that there are, of course, several vendors for supplying the bioreactor where we're supposed to grow the cells from several sources, if it's a bovine, if it's a fish, or if it's a mammary of bovine in order to have a cultured milk. And the whole bioreactor have built-in several biosensors for the temperature and the oxygens and the pH. And the idea of our project and the mission in my lab also is to develop the next generation bioreactor where we will combine a novel biosensors that will detect microbial contamination inside the bioreactor in order to prevent, of course, contamination and to make sure that the scale up of the process, that I think this is the main challenge these days, is how we can make all the knowledge and the expertise that we gained so far in the cellular agriculture field, and how can we take it to the next level where we can do the scalability and to start to produce in a large scale. And this is the very, very challenging step. So the idea of the project is to design novel biosensors that will monitor online or offline. It depends, we will see. So this is one approach, is to develop a diagnostic tool in order to detect the contamination. And from the other side, as Mark mentioned, we need to design non-antibiotic antimicrobial agent in order to tackle the microbial contamination. And the idea of the project with my colleagues from Singapore is to combine peptide, glycan, and lipids also for the mitigation part, but also for the biosensing of the contamination in the bioreactor. So as I mentioned, it's a consortium, it's a great, huge project with Israel, between Israel and Singapore, from the Hebrew University, the Share campus in Singapore, NTU, NUS, and Alliance from Singapore. Nam Jun Cho and Asaf Riedler are the main PI from the two groups, and we have several Singaporean and Israeli partners who take part of this project. So if we would like to design, I will tell you about the diagnostic tools that we are developing. We develop peptide-based bacterial biosensor. Like there are several examples of biosensor, bacterial biosensor, but all of them are not selective towards a specific strain or specific bacteria. They can be like as a spectrophotometer, they can detect the presence, but they cannot tell you what is the type of the contamination. And of course, if we will have something, diagnostic tools that would be accurate and selective, it also will advance also in the clinics and in the health system, so we can detect very precisely if it's a viral or microbial or bacterial contamination. And our approach is basically based on peptide-based biosensor. And in order to design the selective biosensor, we need to have the recognition layer that would be very selective towards one type of bacteria. So we have several examples of the non-selective biosensor in the literature and also in our lab, as we published recently. But I wanted in this talk to tell you about our attempts in order to design selective biosensor. The idea of the approach is basically, we need to look on the surface of the bacteria to look for a specific and unique motif in order to design a selective anchor that can bind it very selectively. So there are two main approaches, the rational design, so we can study protein-protein interaction and the very specific interaction on the bacterial surface. But of course, it takes time and a lot of efforts because you need to develop a specific interaction and study them and then explore the interaction if they valid on the bacterial itself. What we started from, we have also projects like that, but we started from a combinatorial screening, like high-throughput screening, and we used phage display screening for the whole bacterial cell in order to design and to discover selective peptide that will bind the bacteria that we are targeting. So I will tell you in brief, like the process. So the first step is the screening with the phage display library. And the phage display is a library that we change several positions in the capsid protein of the phages. And this position, this is a 12 amino acids that are mostly randomized on the surface of the phage. And then we are looking for the phages that were able to bind the bacteria, the target bacteria, and then we can use next generation sequencing in order to discover the specific sequence that did bind the target bacteria. The next step is the synthesis of the peptide. Then we can explore the interaction using several methods, and then we will immobilize the lead peptide on a gold electrode in order to be able to use electrochemical biosensing. So in order to design this selective biosensor, the main challenge is in this area, the yellow one, the recognition layer. And the recognition layer is the layers that the bacteria are supposed to bind, and we are aiming to have a very selective recognition layer, and this is a very challenging task. So I will share with you our efforts so far. And of course, we are using many types of techniques combining chemistry, biochemistry, macrobiology, biophysics, and other approaches in order to validate that the binding and the process of the development of the biosensor is valid. So for the phage display selection, we are using a library that we designed in the lab, and we are exposing the library to several types of bacteria. So if, for example, for this type of bacteria, the process, we used the Pseudomonas aeruginosa as our target bacteria, and we use several negative selection, other type of bacteria, in order to enhance the selectivity. So in every steps, we are doing, of course, the washing, the elution, and then at the end, we are taking the phages that bind the selective, the target bacteria, and doing the elution and using next generation sequencing and comparing using several bioinformatic approaches the propensity and the frequencies of each of the sequences in each type of the bacteria in order to find the most selective sequence for the target bacteria. And here, for example, it was Pseudomonas aeruginosa. After the sequencing, we can generate many type of sequences. This is just the 3 amino acid, but all of them are 12 amino acid. We are writing an IP right now, so I cannot share with you the full sequence, but you will need to believe me that this is the first 3 amino acid, but there are 9 more amino acid. And you can see that, for example, for Pseudomonas, we have high reads, many sequences that were bound Pseudomonas aeruginosa, much lower binds methicillin-resistant Staph aureus, and few binds the E. coli bacteria. So that means that this is the lead peptide that might have very selective binding propensity, but we need, of course, to take it to the next level, where we synthesize this peptide and label them with fluorescein and explore the binding affinity using flow cytometer when we expose them, incubate them with different type of bacteria. And you can see this is the first peptide MIM, and you can see that it binds very well the Pseudomonas aeruginosa, the green line, but we can see that the E. coli and MRSA still has some percentage of binding. But when we compare it with the different peptide, the second peptide, LTN peptide, you can see that we get higher selectivity. There is a good binding for Pseudomonas aeruginosa, while very low binding was detected for the two other type of bacteria as E. coli and MRSA. So that means that it has high selectivity for the LTN for the target bacteria as Pseudomonas aeruginosa. And the next step in the process, and the most important one, is to immobilize this peptide on a surface, because the idea of the project is to develop a biosensor that would be an electrochemical biosensor, and we need to immobilize the recognition layer on a gold surface using a cysteine and a linker. And the most important and the challenging part is to be able to preserve the binding as we observed in a solution. So we use several techniques and several linkers in order to preserve the LTN peptide binding ability on gold surfaces. And of course, we extensively characterized the gold surfaces and studied the diffusion and the self-assemble on the surface itself. And then we wanted to explore the ability of the different type of bacteria binding to the surfaces. And you can see here, we use a Cyto 9 in order to label the bacteria, expose it to the different type of surfaces. On all of them, there is the LTN peptide, but here we expose it with Pseudomonas aeruginosa for 10 minutes, and this is with E. coli, and this is with Staph aureus, and you can see that we got a very high binding affinity towards Pseudomonas. You can see that it's very, very condensed, and the microbial load is identical in these three bacteria. For E. coli, we got a very low binding, and for Staph, there is binding, but it's much lower than Pseudomonas aeruginosa. So we could detect by eyes, by fluorescent microscopy, the differences in the binding affinities. And the next step that was much more curious for us is to see if we could detect the differences in the binding using electrochemistry, and we're using pediatric electrochemical in pediatric assay. And you can see here that we designed the electrode, the gold electrode, immobilized the LTN peptide, and we could see differences in the normalized RCT in the electrochemical sensing and the differences between the bacterial cells. And we observed high RCT. That means that there is much higher binding for Pseudomonas aeruginosa compared to the two different other bacterial cells. So we are still working on it, and there are many types of electrodes that we are now trying to improve the selectivity, but still we propose this approach as a platform because we use a very generic phage display screening. We can design any bacterial biosensor for any target bacteria. So this is the side of the diagnostic part, and our main mission or vision is to add, in addition to the built-in biosensors, as I mentioned, for oxygen, pH, and temperature, we will add an additional biosensor for the microbial contamination. And if we will be able also to know which type of microbial contamination we deal with, it would be much easier for the system in order to overcome it or to prevent any other contamination. So this is one side of the story. And I would like to tell you also on the other side, and the development of non-antibiotic antimicrobial agent. And as we all aware, also in the clinics, that there is a huge concern about the resistance, antibiotic resistance. So this is from the clinic perspective and the health system perspective, but also as a food scientist, we cannot use antibiotic, not in a regular food system, but also in the cultured meat. There are many regulations about the consumption of antibiotic or the usage of antibiotic, and also in the agriculture field, there is a huge restriction. So the idea is to develop non-antibiotic antimicrobial agent, and of course, it would be also very helpful to know the resistance occurrence toward this new agent. And this is what I will tell you in my second part of my talk. And as a peptide chemist, I was really amazed to see that there is a group of agents, as there are antimicrobial peptides, that we all have them on our skin, eyes, ears, and many, all the organisms on Earth actually has them as a part of the innate immune system, as a shield to prevent contamination. And what for me was amazing to see that there are, of course, many sources, and you can see in this colorful slide that they are really different from one to another. Like there are alpha helical, beta sheet, or even unstructured peptide, and they all possess antimicrobial activity. And for me, it was kind of puzzling, like how come that they are so broad, but still possess the same, induce the same antimicrobial property, mainly disruption of the bacterial membrane. And these peptides were first discovered like 40 years ago, and still there are many open questions in that field. But what we did is to explore what is the main features, the common feature for all of them. Okay, so this is not the sequence, because we can see that this is different sequences, different structure. So what is the common feature? And what we observed in several databases, that they all contain positively charged amino acids, so they will interact with the negatively charged bacterial membrane, and they need to have at least 50% of hydrophobic amino acid in order to tether inside the lipid chain and to possess the mode of action by killing or disruption of the bacteria membrane. So because the differences are really high in terms of sequence and sequences, we decided to change our approach, and we asked if we really need a defined sequences in order to possess antimicrobial property. And the idea was to use the solid phase peptide synthesis, and instead of synthesizing one specific sequence along the peptide chain, we decided to design a combinatorial library of many sequences of peptide, but they all will contain the two important amino acids for antimicrobial activity, and this is the hydrophobic and cationic amino acid. And in each coupling step, instead of incorporating one pure amino acid, we are incorporating two amino acids, and then we are generating the library. So in the first step, if we are using hydrophobic and cationic, we will have two options. In the second step, we will have 4 options. In the third step, we will have 8 optional sequences, 2 in the power of 3. And of course, we can continue as many steps and many chain lengths of the peptides that we are aiming. And in my lab, we're working on these agents for the last 9 years. We explored it extensively, and we showed how the composition, which amino acid we are using, how the chain length of the peptide, and how the stereochemistry will affect the antimicrobial activity. And you can see that at the end, what we observed that the 20-mer amino acid is the optimal chain length. So we are generating 1 million of peptides in the pool, in the mixture, that all of them contain the same 2 amino acid, but the order of the 2 amino acid along the chain length is variable and randomized between one of them and another. At the end of the synthesis, we cleave the peptide from the solid support very easily in acidic condition, and we are generating a free in solution peptide antimicrobial mixture. When we explored the antimicrobial activity, we were really amazed to observe that we have very broad antimicrobial activity. This is the minimal inhibition concentration, and this means that what is the exact concentrations that is needed in order to prevent the growth of the bacteria. And we have here gram-positive and gram-negative bacteria, and also clinically isolated bacteria. And here, for example, you can see that the leucine-lysine mixture have very good antimicrobial activity towards gram-negative and gram-positive. As you see that the values are really low, that means that the compound is very active and prevent the growth in a very lower concentration. In collaboration with Max Riandov from NPL in UK, we characterized the mode of action, and here we were amazed to see that on using AFM, atomic force microscopy, where we immobilized on the mica a lipid bilayer that mainly negatively charged in order to mimic the bacterial membrane, and we incubate our phenylalanine lysine 20-mer mixture, and you can see very nicely the pore forming that are really uniform in diameter. It's a 200 nanometer diameter. You can see the self-assembly process here, the tail, that are actually concentrated inside the pores. As a complementary method, we use the secondary ion mass spectrometry, and this is the reason why we use the N15 isotopically labeled phenylalanine. And you can see that all the peptides are really concentrated in the pores, and we propose that this is the mode of action of the peptide also on the bacterial membrane itself. Here we label the peptide with fluorescein in order to visualize the penetration into the bacterial cells. You can see very nicely that they penetrate very, very fast. Even after 1 minute of incubation, we can very easily see and detect the peptide inside the bacteria. And they might have also other role inside the bacterial membrane, but not just. So we are currently working on that. We also studied how this agent can eradicate and degrade mature biofilm, which is a real threat also in the food industry and also in all the bioreactors that we are working with. And the idea is that the bacteria replicate themselves and extract an extracellular matrix, so it will be very isolated from the environment. And then all the antibiotic treatment is not so effective, and it's very hard to treat this mature biofilm, especially because the bacteria inside the biofilm are in a growth arrest, and then all the conventional antibiotic is not active anymore because all the processes are really shut down in these processes. So what we did, we used methicillin-resistant Staph aureus, and we showed very nicely how we can take mature biofilm, use our agent, and degrade more than 50 or 80% using 25 microgram per ml. This is the biofilm biomass using crystal violet. And also we did the scanning electron microscopy to visualize the degradation and the eradication of the mature biofilm very easily. We developed also several new generation of the antimicrobial peptide. I will skip it. I will just mention that we were able to shorten the chain length of the peptide from 20-mer peptide to 5 amino acid with a palmitic acid, with a fatty acid, in order to keep the balance between the hydrophobic and the cationic proportion. And we studied extensively all the sequences and all the peptide, but I think this is for another talk. But what I can tell you, what we studied in this system is, because it's just 32 peptide in the mixture, because it's 2 in the power of 5, we were able to synthesize each one of the sequences and study more deeply the interaction between the peptide. Because when we synthesize 1 million of peptide the sequences, we observed great antimicrobial activity, and we also wanted to explore the interaction between them. And in that system, we were able to see that there is a synergistic effect between the active peptide in the mixture, and then they enhance the antimicrobial activity. And if you would like to see, we published a paper a few years ago where we characterize all the synergistic and the values of the synergistic between them. But I think the most advantage of our approach that, okay, they are really active. It's very easy and cheap to synthesize, so we can imagine that we can use them as a crop protection, as a food preservative, and also for the cellular agriculture industry. And there is a synergistic effect as we showed in the 5-mer sequences. But our main hypothesis is that because it's a mixture and a cocktail of compound, it would be very difficult for the bacteria to develop resistance toward this cocktail of peptide. So what we did recently, Bar Maron, very talented PhD student in my lab, we established an experimental evolution assay, where we expose the bacteria every day for a fresh media with the active agent, and we screen it and cycle for a month or even more, and we monitor the sensitivity of the compound along the assays, and this is the experimental evolution. And as a comparison, of course, we can take antibiotic, but it's not so challenging, because for antibiotic, we observed high resistance occurrence even after few days. So we selected several antimicrobial peptides with specific sequences, and we compare the rate of resistance occurrence compared to the specific sequences of antimicrobial peptide as melittin, pexiganan, and temporin. And this is the summary of the result of the experimental evolution after 30 days of cyclings, of many cycles of the experimental evolution. And what we can see that for melittin, we observed a high resistance occurrence. That means that at the end of the assay, at the end of the experiment, we needed to use 30 times more concentration of the peptide in order to prevent the growth of the bacteria. And for pexiganan, the same, but 10 times more, 10-fold more. For temporin, it was very low, but for the random peptide mixture, for the phenylalanine mixture, as you can see here, we could not detect any differences in the activity. That means that there is no resistance to the random peptide mixture. And of course, we are really interested in understanding how come, and we also studied the cost and all the other parameters in the experimental evolution, how did it affect the resistance occurrence to one peptide to the mixture, or how the exposure to the random peptide mixture for 1 month affect the resistance or the activity towards other type of peptide. It's also very interesting. Now we are doing like whole genome sequencing in order to better understand the mechanism of the bacteria, how they protect from melittin, but how they cannot develop resistance toward the random peptide mixture. And I hope that we will have more data about it in the near future. The next step was to study if this non-antibiotic antimicrobial agent have also activity as an in system, like efficacy in mice models. And of course, we needed first to do a lot of safety study, also with red blood cells, with cell lines, and in several mouse. And you can see that we could not detect any toxicity effect in all the assays, and also in the mouse models that we did it in collaboration with Guy Lau from USA, from University of Illinois. And then we established several methods and assays to explore the efficacy also. So they are safe to use in mouse, but we wanted also to study the efficacy. So we developed several assays, several models in mouse for pneumonia, with Pseudomonas aeruginosa or with methicillin-resistant Staph aureus, and bacteremia with the same type of bacteria, Pseudomonas and MRSA. And after the treatment of 5 milligram per kilogram, we can see a great reduction in the bacterial load, if it's in the lung or in the spleen, depends if it's bacteria or pneumonia. And we were really encouraged by this result because this is compared to a very low concentration of the random peptide mixture. We also advanced the study where we decided to work on Acinetobacter baumannii, which is a very nasty bacteria that causes a lot of death in hospitals. And you can see the survival rate of the mouse that were treated with the random peptide mixture were almost 100%, while the control, after 50 hours, most of the mouse were died because the disease severity was very high. And this is very encouraging results also in the area of the non-antibiotic for systemic use. So as I mentioned, we're working extensively on these agents, and we worked on them in water treatment. We developed several beads that you can use as a source for prevent contamination in water or treat microbial contaminated water, and to make them pure for use. We use them as a crop protection agent in the field. We can spray them in the greenhouse in order to prevent the disease severity of plant pathogenic bacteria. We use them as a food preservatives in several systems as minced turkey meatballs. We are really encouraged and interested in developing bioactive packaging. And as I mentioned now, I showed you the antibiotics, and you will hear soon by a very talented student from my lab, Idani Akir. He's a master student that will tell you how we took this agent and we are exploring them, the use of them in the cultured meat industry. And I will leave it for Idan to tell you more about it. So this is the overall goal. From one side, the diagnostic, and from the other side is the mitigation. As I told you, it's shared as a joint project. We join forces with Singapore, and I hope that soon we will have more update, and I will share with you next year our result. In addition, we 2 years ago, we established a spin-off company from my lab based on the random peptide mixture, the company called Prevera, and their main mission is to take this technology and to make it industrial. So they have a lot of work on regulation and scalability, but they are doing a really job, and we have a great progress also in the regulation and the scalability, and also in the cultured meat industry as well. So if you would like, you can have a look in their website and to learn more about it. And I would like, of course, to thank my group members. Without them, of course, they did most of the job. This is Idan. We'll tell you in short, like the process, our efforts in the cultured meat, Einav, Aya, Bar for experimental evolution. And I would like to thank them, and I would like to thank the funding, and I would like to thank you for your attention, and you, Mark, for the opportunity. And if you have any question, I will be more than happy to answer. I have no idea, but thank you about the timing, but I hope I'm okay with time. Thank you very much.
Well, thank you for giving this wonderful lecture in these troubling times, that you find the concentration to do that. Was really grateful for that. We have time for one pressing question. I'm sure you have tons of them, but who wants to have the honor over there? For the people who are joining online, you can also ask questions through the chat, and then they will be translated here and expressed here from the people from the team. So please feel free to do that.
Yes. Thank you very much for this interesting talk. has been mentioned that these antimicrobial peptides that have been synthesized, they can be used as food preservatives. So I was wondering, what is the resistance of this peptide polymers to food processing conditions like pasteurization or acid hydrolysis in the guts of humans or animals, or proteolysis by proteases by other microorganisms in a soil environment, in a plant environment? Thank you.
Yeah. Okay, thank you. That's a very good question. And as a peptide chemist, we can change the amino acid to non-natural amino acid, and then we can generate a proteolysis proof, resistance proof, like they cannot be digested by proteases. But I think in this direction, as a food preservative, we want it to be totally degraded in our gastric system. So we explored the activity also in mouse model, and you can see that natural amino acid, the L amino acid, are really active, even though it was injected to the blood. So I think it's something that we will need to balance between them. And also in the project of the turkey meatball, we're in the minced turkey meatball, we could show that we can extend the shelf life of the turkey meatballs in 8 days, but just by incorporating our agents. So that means that they are still active, but I'm sure that they are being proteolytic digested. And we can monitor and play with the ratio between it with the non-natural amino acids that we can add to the mixture. But I think the most advanced and the advantage of our approach is to use the L amino acids, that they will be as a food, in the food, they will keep the food non-contaminated, prevent the contamination. But when we consume it, they will be totally digested, and then the amino acid would be used for other protein synthesis. So I think this is the idea. And also it's important not to change the microbiome of the consumers. And this is also aspect that we studied in the lab. So as long as we are using natural amino acid, the L amino acid, the regular amino acid, we could not detect any microbiome differences. But when we use non-natural amino acid for the synthesis of the random peptide, we could observe changes in the microbiome. So I think it's something that we should to keep in mind.
Thank you. And once again, thanks a lot for joining online, and have a great day. Thank you.
Thank you for inviting me.